Hello everybody, super happy to see all of you. Very good evening. This is your Shruthi Maam and today I'm going to revise the entire human physiology for you. But now when you're studying, right? Now this is not the first time that we are studying, correct? We've already studied, we've already written an exam and this is a re-exam that we are going for. Now at this point in time, recollecting all of the facts and trying to retain them can be twice as hard, which is why I have brought a very interesting tool. Now this is an app that is available to study the anatomy and physiology of the human body. I have taken snippets from this particular app to give you like a live representation of how things look. So now what's going to happen, you're going to have a visual memory, a visual capture of the concepts of the different parts of the human physiology, the different mechanisms that are going to happen. So now this is going to be easier for you all to retain and easier for all of you to understand the concepts. Okay, now for those of you who did not get enough time to do human physiology the last time, please make use of this particular opportunity and thorough your human physiology, okay? Because I personally feel like the more you're strengthening your human physiology, the more you are very confident. 6 chapters are worth almost 50 questions, okay? Easily 50 questions. So what we expect is not 50 questions, I mean marks, right? Almost 50 marks of your paper will come from the human physiology. So roughly you can expect at least 2 to 3 questions from each of the chapters, means 12 to 18 questions easily. Okay, so that accounts to about 50 to 60 marks in your paper. Okay, so that is pretty much a very good amount for all of you, right? Especially when you are targeting to boost your rank, and especially when you are willing to take that extra step to increase your score and go from that comfort range. Definitely these are the chapters that you will have to work on. Is that okay? Raghav, everyone, so definitely do pull in your friends as well, and let's do the ultimate revision today. Okay, so I'm going to start with breathing and exchange, and we're going to go all through the 6 chapters of human physiology with examples, with representations, flowcharts, and super simple for all of you to understand. Okay, now to get all of these notes, I'm going to be sharing it in my Telegram group. The link is there in the description box, so definitely do come in and check out the Telegram group. Is that okay? So all of you all will get the notes, will get everything you need for your preparation. And also, I am doing a study with me challenge. So every single day I'll be posting your daily revision chapters and a small test for it. And I hope all of you are going to keep up with the challenge. Okay, so I want to make sure that all of you are finishing the challenge to the end, so definitely do join my Telegram group and tune in for the challenge as well. Okay, so a lot of you told me that, ma'am, I don't have Telegram, so what do I do? Where do I get the notes? Right now what happens is, if you use any other form of media, once you join the group, you may not be able to see the previous messages. Okay, so here in Telegram, no matter when you join, you will be getting the access to all the different content that I've already posted on the group. So definitely install. It's only one app away. Okay, we install so many different things into our phones. So just install this one app and use it exclusively for your study purpose. You'll be getting the notes and all the notifications there. So it will be super useful for you. Okay. Okay, that's lovely, Raghav. But as we are preparing for the NEET exam, I want all of you to have the same seriousness that you had for the actual NEET exam on May 3rd. Okay, now no matter what happens in the NEET exam, also we must target a 360 on 360 no matter what. Okay, no excuses, nothing. All right, so throw some fire, throw some stars in the chat box, and let's start human physiology, shall we? Yes, everybody. Raghav, Kanishka, let's go. Yes, Kanishka, let us go. Okay, Yes, so the first chapter we have is breathing and exchange of gases. Okay, now for breathing and exchange of gases, what all should you know in the chapter? First, the respiratory organs, okay, the gross anatomy of the system, then mechanism of breathing, how the muscles are moving, how the pressure is changing and how breathing is happening, how the exchange. Now once you are breathing, you're taking in air from the outside deep into your lungs, correct? Now from your lungs, it has to undergo an exchange into the blood, and this blood has to carry the gas to the different parts of the body. So that is where we're going to look at exchange of the gas and then also the transport of the gases, okay, and how this entire system is regulated, followed by the disorders. This is all you need to know in this particular chapter, okay? Now let's start off. Firstly, when you look at the human respiratory system, okay, this is how your actual respiratory system would look. It starts with a nostril, okay, and from your nostrils, so I'm just going to label it as I am going to see. So these are my nostrils, okay. Now from the nostrils, you're going to have a nasal chamber. Okay, now this nasal chamber is the gap behind your nostril. It's the chamber inside your nose. Okay, now from there, you're going to get to this huge tube-like structure called as the pharynx. Okay, now this pharynx extends all the way from the nose up until your throat. Okay, and from this throat region or your pharynx, it moves into this cone-like structure called as the larynx. Now this is where the voice is generated. The voice cords are present here, followed by the trachea. Okay, so from the pharynx we have the larynx, and then from the larynx we have the trachea. Now from the trachea, these tubules are going to branch and enter the structure of what we call as the lungs. Okay, now these lungs, though a huge organ, are made up of small sac-like structures called as what? The alveoli. Okay, now this alveoli together make up the lungs, and around the lungs, now when you buy a new sofa, like how you have a wrapping paper around it, a plastic cover to make sure that nothing is damaged, same way every organ in the body also has a certain wrapping paper or a certain covering to it. Okay, now this covering here is called as the pleura. Okay, now this pleura is double membraned. One is touching the lung directly and one is facing to the outside. Now in between this pleura, we have something called as the pleural fluid. Okay, now this pleural fluid is responsible for lubricating the surface and shock absorbing and trying to protect no matter what happens. Okay, so there is a fluid filled cavity inside, and there is a fluid that is helping the lungs to not get damaged so easily. Okay, now this pleura is the covering of the lungs, remember that. Okay, now when you look at the tree, right from your nostril over here, we have a nasal chamber over here. Okay, and then from the nasal chamber, it comes down to the pharynx. Now can you all see here? We have a clear cut pipe starting from the nose through the mouth and then through the larynx as well. So it's a single tube that is continuing. Now this is the larynx. Now sometimes you have seen in some men, they have a very prominent structure up in the throat, right? Now that is nothing but the Adam's apple, they say, which is a protruding part of the larynx itself. Now inside the larynx, you can see there are vocal cords which when vibrated will give rise to the actual sound. Okay, now what happens? Now we have a single tract that is acting as, from the nose also, from the mouth also, we have a same connection. So that means when you're eating food also, it has to pass through the same pipe. So here what happens is, when the food is coming down into the pharynx, in order to make sure that this food does not fall off into the larynx, but it should go behind into the esophagus. Okay, my esophagus will be here. So in order to help with that, there is a small flap-like structure which we call as the epiglottis. Now remember that this initial portion, right, the starting part of the larynx, just above the larynx, is what we call as the glottis, and this glottis is covered by the epiglottis to make sure food does not pass through when we are swallowing the food. Okay, fine. So now coming into the larynx, just below the larynx, we have what we call as the trachea. Okay, now this trachea is having some, as you all can see, some cartilaginous rings around it. Okay, so it has incomplete rings, incomplete cartilaginous rings. Okay, now why is this? To make sure that the trachea will not collapse. Okay, now why should it collapse at the first place? That is because, now see, when air is not there in a balloon also, patak, it will fall off. Correct. So in order to make sure that the sac-like structure is maintained and there is no problem, we are going to see that the cartilaginous ring will hold the trachea even after the air is released off. Okay, is that clear, everybody? Yes, good. Okay, so here if you see, these cartilaginous ring make sure that the trachea does not collapse when the air is gone. Okay, now from the trachea, if you see, there is further branching. Okay, now from the trachea, we have bronchi entering the left and the right lung. Now from there we have further primary bronchi, secondary bronchi. Okay, so we have primary, secondary, tertiary. Then we have bronchioles, which are further tinier structures of the bronchi. Okay, and like these bronchioles divide into terminal bronchioles, and this leads into the alveolar duct. Now, until this part, everything is only the conducting part. Okay, there is absolutely no exchange that's happening here. So it is only a conducting portion of the system. Okay, now the alveolar duct, if you see, this is the part that leads into a sac-like structure called the alveoli. Okay, now if you look at the alveoli, as you can see here, these are small sac-like structures that are present. Okay, now these alveoli have very thin squamous epithelium. Okay, that is comprising the wall surface. Now this squamous epithelium is going to make sure that the layer is so thin that diffusion can automatically happen very easily. Okay, is that clear? So the whole part until the alveolar duct, I said, is majorly a conducting part. Now this is the only part that is involved in the exchange of gases. Okay, now this is highly vascularized, as you all can see here itself. So there is extreme supply of blood, there is extreme supply of a lot of vessels there. So the blood can come in contact with this diffusion membrane, it can diffuse the gases and it can take the respective gases and go back inside. Okay, so this process is extremely made simple because the alveoli membrane is super thin and supports the diffusion very easily. Clear, everybody? So the major difference between conducting part and exchanging part is very important. Until the alveolar duct, nobody is doing any kind of conducting. From the alveolar duct, the alveoli is the only place where the actual exchange is happening. Okay, clear? Kanishka, Abhinay, Raghav, RJ. Yes, RJ, I've promised I'll complete all the 6 chapters of Human Physiology. Okay, that's what we're going to do in today's class. Clear? Right, so now that we're done with the gross anatomy of our respiratory system, we'll move into the mechanism of breathing. Okay, so in order to let the air come inside and go outside, we have a set of movements that is carried out by the ribs, the muscles in the body, and this in turn changes some pressure in the body. I'll tell you how. So simply, if you see this particular muscles in between, okay, as you all can see, is what we call as the intercostal muscles. Okay, now these intercostal muscles are present between the ribs. And then we have one dome-shaped muscle called as the diaphragm. Okay, so when you have the space inside your lungs, which is very, very small, very, very less, what happens? You have only small number of people who can sit in that space. Now, if you increase the space, lot of people can come and sit. Now, if you decrease the space, if it is too congested, everybody has to move out. It is the same logic that is applied for the mechanism of breathing as well. Okay. So, during inspiration, what happens is, first, the intercostal muscles as well as the diaphragm is going to contract. Okay. So, once the contraction is happening, what happens? It's going to extend the contraction is going to take place, and as a result, the volume of my lungs, okay, which is the thoracic volume, is going to increase. Okay. Now, if the volume increases, means more places there is no congestion at all in between. So, that means what? The pressure will decrease. Now, if the pressure is low, but a lot of people are waiting outside, what happens? They can come inside and sit. So, that's exactly what happens, and the air moves inside. Okay, that's about it. Now, when you look at expiration, what happens in the case of expiration? It is going to be the opposite. Now, the intercostal muscles as well as the diaphragm is now going to relax. Okay. Now, once they are relaxed, they are brought back to the original place. Now, they had increased the volume. Now, they're coming back to the original, means the thoracic volume is now going to decrease. So, if the volume suddenly decreased, too many people are there inside. So, as a result, what happens? The pressure will increase. Now, to get rid of that pressure, I need to push the air out. So, I have a expiration that is happening. Okay. Right. Yes, so Abhinay, that's exactly correct. So the conducting part will transport the atmospheric air. So whatever you have, all this from your nose down into your throat up until the bronchial terminal bronchioles, it is only going to carry the air inside, right? After it goes inside, the actual the exchange will take place only in the alveoli. Okay, fine. Now, okay, so RJ 2007, what happens is, see, between the ribs, we have one layer that is outside and one layer that is inward, okay. So that external muscles are called as external intercostal muscles, and the inside ones are called as the internal intercostal muscles. Now, both of them will contract, and as the result of their contraction, what happens is, the muscles are pulled, the rib cage is brought upwards and outwards, and that increases the thoracic volume, and as a result of which pressure fluctuation will lead to the movement of the air. Okay, is that clear? So with respect to your NEET exam, just remember that the volume will increase, pressure will drop, so air will come inside. Now, volume will decrease because of the relaxation, pressure will increase, and the air will go outside. That's it, you need to remember. Okay, now talking about the exchange of the gases, is that clear? Great, Kanishka. Okay, so now when looking at the exchange of the gases, okay, we all know that the alveoli is going to be present. Now, this alveoli has a very thin diffusion membrane, I said. Correct? Now, what is this diffusion membrane made up of? So, first we have the epithelium of the alveoli, okay? Then we have the endothelium of the blood vessel, okay, of the blood vessel, and in between we have some basement membrane. Now, through this only the gases have to reach the blood. So, that means the thinner this is, the better it is. Understood? Now, also the solubility will matter. Okay. Now, what do I mean by solubility? Let's say I have 1 cup. Okay. Now, in that 1 cup, if I can fit only 2 molecules, then I can fit only 2 molecules. But in the same cup, if I can fit 20 molecules, then what? Then it means that I can fit 10 times the previous kind of molecule. So, let's say I have A and B. Okay, I can fit only 2 of A, but I can fit 20 of B. Means the amount of molecules of that particular the substance that I can carry is much more. So that also affects the exchange. Okay, so solubility, for example, of carbon dioxide is much more than that of oxygen. So that is why if you see, the difference in partial pressure will be very less in the case of carbon dioxide, but the difference will be much more when you look at the oxygen. Clear? Is that clear, everybody? Yes, I will teach you respiratory volumes, RJ 2007. Don't worry. Okay. Fantastic. Okay. So this exchange of gas is extremely dependent on something called as the partial pressure. Okay, now what is this partial pressure? Means what is the pressure contribution of CO2 and what is the pressure contribution of O2. Now based on this relationship only, my exchange is going to happen, my transport is going to happen, everything is going to happen. Okay, is that clear to all of you? Fantastic. So where are the stars and the fire in the chat box, man? I need superb energy in the chat box. Okay, we're going to finish 6 chapters, thorough revision today, and that's not simple. Okay, is that clear, everyone? Fantastic. Okay, do subscribe the channel if you have not, and in the process of preparing for NEET, I'm going to make sure that you are clearing all your doubts and you're going to make a fantastic job in your NEET exam. All right, everybody? Fantastic. Now, coming to the transportation. Okay, now if you look at the transportation of oxygen and carbon dioxide, they are completely different. Okay, now if you look at oxygen, we have a molecule known as hemoglobin. Okay, so this is the molecule that's going to carry the oxygen. It's going to convert to something called as oxyhemoglobin. It is reversible, so it will bind and then it will dissociate. Okay, now this hemoglobin, one molecule can carry up to 4 molecules of oxygen with it. Okay, now this particular hemoglobin molecule will bind to the oxygen and take it. So almost 97% of my oxygen is carried into the hemoglobin, and only 3% is through plasma. Okay, now this 97%, for it to associate, what is required? If a lot of oxygen molecules are there, once they see the jeep, they will come and sit inside. Correct? Now they are coming and sitting inside, means inside the car there will be AC, so temperature will be low. Okay, and everything is good, everything is sweet. Oxygen is a good molecule, so there is no acidity, no hydrogen ions, no nothing that is going to affect it. Okay, but if this oxyhemoglobin has to dissociate, okay, for the dissociation to take place, what should be there? Very less of oxygen should be there. A lot of carbon dioxide should be there, so that this oxygen will get down and will start fighting. Okay, so not exactly fighting, just think of a jeep and all that. So that's why I'm giving this kind of an example. So when there is a lot of CO2 outside, I can't be sitting and chilling inside. Correct? So I'll get out and go out. So less of carbon dioxide should be there. Now if carbon dioxide is there, automatically means what? Carbon dioxide is something our body does not want. So that's like the villain. Okay, so if that is the villain and oxygen is the hero, what happens? When there is more carbon dioxide, more villains outside, hero will get down and fight. So a lot of partial pressure of carbon dioxide will result in dissociation. Second thing, if carbon dioxide is there, then so much of heat is there, so much of fight has to happen, so higher temperature will be there. Okay, and along with higher temperature, there will be higher concentration of hydrogen ions, and there will be a lot of acidity. pH will be very, very low. Okay, now under all these circumstances, what will happen? The dissociation of the O2 will take place. Now if you have to remember for association, don't remember 2 different cases. Remember dissociation and just remember it is the opposite. So if you know dissociation very clearly, the opposite is association. Because if you learn higher CO2, lower CO2, high temperature, low temperature, you will get confused. Okay, so just imagine a clear-cut scenario and just remember only that. Okay, is that clear to you? Now, if you look at this, how this oxygen and hemoglobin are getting paired and how they're getting dissociated, we have a dissociation curve that is there, okay, which is basically looking like a curve. Okay, it's like a sigmoid curve. Now this is the diagram is there in your textbook, but there is nothing very important. You just have to remember that this curve demonstrates the dissociation of the oxygen. So under what conditions it happens, I've already told you. So these conditions are the most important there. Okay, is that clear? Okay, now when you come to CO2, it's slightly different. Only 20 to 25% of carbon dioxide will travel with the hemoglobin as a carbaminohemoglobin. Remember, it is not carboxyhemoglobin. Carboxy is with carbon monoxide. Carbon dioxide is only carbaminohemoglobin. The remaining 70% will travel as bicarbonate ions. How? Once the CO2 comes along with the water, it will combine to form carbonic acid. And who is helping with that? Carbonic anhydrase, who is present in the RBC. So RBC will be like, bro, in my car I don't have place da. So you get down and walk. What I'll do is I'll give you some enzymes to help. So he will convert this carbonic acid into bicarbonate ions and hydrogen ions, which is why when there is a lot of CO2, there is a lot of hydrogen ions also. Okay, so that indirectly means that lot of hydrogens are present. Okay, so RJ 2007, mostly you can expect questions on conditions that will lead to the dissociation or association, not directly from the graph. Okay, is that clear to all of you? So once this this molecule has dissociated, next what will happen? It will start going in the plasma, and it will just come through along like that, and about 7% comes as dissolved form in the plasma itself. Okay, now these bicarbonate ions will come to the destination, and once they have to go out, again they'll use this particular enzyme called carbonic anhydrase, and they will convert back into CO2 and H2O, and they will leave the body. Is that clear to all of you? Yes, everyone? Is that clear to all of you? That's it. Okay, so for every 100 ml of blood, okay, for every 100 ml of blood, we carry 4 ml of oxygen and 5 ml of carbon dioxide. Is it correct? Is it correct? RJ 2007, Raghav, Kanishka, Abhinay. It's correct. Yes, everybody? Clear? Yes, RJ 2007, 70% is transported as carbonate ion. See, yes in the plasma, but when you say dissolved in the plasma, it means something else. Okay, so these ions will directly travel. Finally there is an answer. Yes. No, it is not this. Okay, I am just testing if you all actually understood or not. Or you remember or not, more like. So every 100 ml will contain 5 ml of O2, but only 4 ml of carbon dioxide. Okay, please remember that. All right, everybody? Clear to all of you? Fantastic. Okay, so now coming to the most important part of the chapter, but yet the most confused and, ayo ma'am, this question, like that kind of a feeling for all of you, that is basically your respiratory volumes. Okay. Now for respiratory volumes, let me clearly tell you, remember it like a slab. Okay. So first we have the 4 slabs like this. Okay. So one is the TV or the tidal volume. Now what is this tidal volume? Every day what normally you intake and exhale. Okay. Inspire, expire. Normally when you're not doing anything, when you're just, you're just breathing, you're just watching the class, you're just watching TV, you're just sitting and doing your own work. So that time the normal amount of air that you breathe in and breathe out is the tidal volume. Now, if you decide that you want to take up more air, then what is the remaining space that is available for you to inhale? To take a one deep breath, It is the inspiratory reserve volume, which is roughly 2500 to 3000 ml. Okay. Now, when you look at breathing out, same thing. Now, normally this is how much you will breathe out. But if you have to push yourself and be like, oh boy, I'm going to let go of all the air in my lung, then that is the expiratory reserve volume, which is close to about 1000 to 1100 ml. Okay. Now, beyond that, even if you want to push out, you can't push out this remaining little bit of air present in the alveoli, which we call as the residual volume. This is close to about 1100 to 1200 ml. Okay, now this particular residual volume is the reason even after you breathe out, you will not be completely having a collapsed lung, or it will not, the walls will not attack and collapse on itself. The little bit of air will make sure that the lung will maintain its shape. Understood? Now when you look at the capacity, what did I say? Tidal volume is what you will normally breathe in, breathe out. What is the total capacity that you can inspire, which is normally how much you can take? I'll use a different color. Okay, normally how much you can breathe plus how much you can forcefully breathe. So this becomes your inspiratory capacity. Okay, now the opposite also counts. Now if this is normally how much you will exhale and you will leave out, then this together with how much you can forcefully do is the total volume you can expire. Right? That's it. Okay, and when I am normally breathing, okay, when I am normally breathing, how much is the residual air that is inside? One that I can forcefully push out, but I am not pushing it, right? It is still there in my lung. That plus what will never go out together is the residual amount that is present when I am normally functioning, which we call as the functional residual capacity. Clear, everybody? Now this total IRV plus TV plus ERV, totally how much I can take in and I can completely let go is what I call as the vital capacity, and including the residual volume, everything together is called as my total lung capacity. Now only remember these 4 slabs and these 4 amounts. You don't have to remember anything else, because once you know what each of these terminologies mean, easily you can calculate from there. Okay, is that clear? Is that clear, everybody? Superb. Okay, so this respiratory volume part will definitely make a difference, and definitely one question you can expect to come from this portion. So be careful. Now sometimes they can ask you some numericals. They can say residual volume is so much, TV is so much, IRV is so much. Simply don't use all the numbers that they've given you. Okay, look at what exactly they're asking you and only calculate that, because sometimes they'll give you unnecessarily one word, one terminology. So it is very important for you to understand what exactly you need to do and what exactly you need to calculate. Okay, so for this, respiratory volume is very, very important. Now coming to the regulation. Yes, I inspired through my lungs. I understood how my breathing is happening. I understood how exchange is happening, and I understood how the transport will take place for the oxygen as well as the carbon dioxide. Now, how am I going to regulate this? Firstly, remember that we have 2 centers. Okay, one is the respiratory center in the pons, respiratory center in the medulla. Okay, in the pons we call it as the pneumotaxic center. Okay, so pneumotaxic center in the pons. Now both of them will make sure that I can adjust the time duration that I am inspiring and expiring. Now let's say I want more air, I need to quickly take in multiple breaths, right? And if I want less air, I can slow down my rate in such a way that I can control how much of air I am taking in. So both of them work together, and they will make sure that the duration of inspiration and expiration can be adjusted in order to accommodate for the changes. Okay, now also we have certain chemoreceptors. Okay, we have certain chemoreceptors that are present especially near the carotid arch, near the central nervous system. They keep looking out for the carbon dioxide molecules. If they see that too much carbon dioxide is there or too much of hydrogen ions are there, they will immediately tell the brain. The brain will call the medulla or the pons. They'll approach the pneumotaxic center. They will adjust and make sure that this excess carbon dioxide is thrown out by adjusting my intervals of my expiration and inspiration. Is that clear, all of you? That's about it. And the last part of this chapter is the disorders. So when you look at disorders, simply two important disorders are there. Firstly, it is asthma. Okay. Now, asthma is an allergic reaction. Okay. And this is caused because of inflammation in the bronchi, in the trachea. Okay, so those things can cause the difficulty in breathing, but it's only an allergic reaction, so drugs administered can control the disorder. Okay, the other disorder you need to know about is emphysema. Now, emphysema is where the surface of my alveoli is damaged. Okay, so because it is damaged, that means decreased surface area for exchange. Correct? So as a result of which there is difficulty in breathing. Cigarette smoking is the top reason why this can happen. Okay, and this will grow over time. Okay, so it's a chronic disease. All right. Now, apart from that, we have something called as occupational disorders. Occupational disorders. Okay, so now these are because of where you're working. Okay, let's say you're working in a mining place or somewhere where there is extreme exposure of minute particles like asbestos, silicones and all that. So there, because you inhale these particles over a long period of time because of your profession, you end up getting up these diseases, which we call as occupational disorders. That's it, and we're done with the respiratory system. The breathing and exchange of gases is done, guys. After I do one full round of revision, I am going to plan classes for your PYQ practice, for a lot of discussions as well. So definitely subscribe and stay tuned. Okay. Okay. Reduce the duration of inspiration. Okay. So RJ 2007, what that question really means. Okay. Understand that once the air comes in, there is no choice but to push it out. Correct? You have to push it out. Now, depending upon how much oxygen is there, we don't regulate our system. Depending upon if there is a lot of carbon dioxide is when all of these regulatory systems will get triggered. So, our goal is to throw out the remaining carbon dioxide. Correct? So, if I change how much I inspire, I can change how much I expire. Okay, so what comes in is the first. That is the active process. Once it goes out, going out will happen on its own. You don't have to spend any energy on it. Okay. So simply when you're breathing also, if you want to forcefully breathe in, you need to make some effort. Okay. But once you hold your breath, do you need to make effort to put that this thing outside? No, you don't have to make any effort for the air to move outside, right? So that is why you have to, reduce the duration or change the duration of inspiration in order to control the regulation of your respiratory system. Okay? Clear, RJ 2007? I hope it's clear. Okay. Perfect. Any other questions, anybody? Shall we move on to body fluids and circulation? Now, let me remind you that I am doing the Study With Me challenge. So, every day I'll be sending you a portion and a small test to do. So, definitely do tune in because it's going to be very, very, very useful for you. Okay, everybody. So, if you've not subscribed yet, definitely do subscribe. And let's move on to the next chapter, shall we? Fantastic energy, Kanishka, RJ, Alan Entertainment. Okay. RJ, just to make sure everybody can understand, I'm doing this, so I hope you will follow through the class. Okay. Perfect. Okay. So when you look at body fluids and circulation, now that we know how the air is coming in and going, we need to also understand how the transport and circulation of all of these items is happening. RJ, yes, so fibrosis is nothing but there is too much of proliferation of the tissues inside the lungs, and that blocks your pathway, that will allow some, inconvenience while breathing, and that's what you call as fibrosis. Now this fibrosis not necessarily in the lung, it can happen in any part of your body also. Okay? Yes, Kanishka, daily there will be classes. Okay, now let me remind you, tomorrow morning I will be coming live again to discuss a very, very important unit. We'll be finishing up genetics tomorrow. So morning, tune in for the class, okay, and it will be super useful for you. Don't forget, okay. So after today's class, don't run away, and we are not done with today's class. We have 6 to go. In that, we have already finished 1. So 5 more to go. All right, everyone. Okay, now when you look at body fluids and circulation, like I said, we're talking about how the circulation is going to happen in the body. Okay, now when you look at blood, blood is what? 55% of plasma and 45% of formed elements. Okay, now in your 55% plasma, what do you have? We have 90% only water. Okay, so water is definitely there. In the remaining portion, what do we have? We have 3 important proteins. Okay, so the first protein we have is the globulins. Okay, so globulins are nothing but your immunoglobulins, your defense cells. Okay, then apart from that, we have the albumins. Okay, now albumins work for your balance of pH and balance of the salts. So osmoregulation is involved because of the albumins. Okay, and then we have another protein called the fibrins. Okay, so fibrins are nothing but these are proteins involved in the coagulation of blood, in the clotting mechanism of the blood. Okay, so I will talk to you about the clotting mechanism as well. So remember that fibrins are a very important protein that is present in the plasma. Okay, then in the remaining 45% of formed elements, we have RBC, we have WBC, and we have the thrombocytes or the platelets. RBCs are also called as erythrocytes, WBCs are also called as the leukocytes, and platelets are also called as the thrombocytes. Okay. Now this RBC is the most abundant blood cell, and that is why you see your blood in red color. Okay, simple as that. Now this RBC is the one that will decide your blood grouping. Okay, so here let me tell you that the blood can be grouped into the A, B, O categories. So if the RBC has A antigen, it is A group. If it has B antigen, it is B group. If it does not have any antigen, it is O group. Okay, but A and B together, if they are present, you can have an AB group. Okay, depending upon what antigen you have, you need to give the same kind of blood to the patient. Otherwise, what will happen is there will be some immune response that is triggered, and that will cause your entire, even it can cause death. Okay, so that will be some dishum happening inside your blood. So you have to be very, very careful while you are grouping the blood and giving the blood to the patient. Okay. Okay, so RJ, you don't have to remember all the minerals, just remember the 3 types of proteins and roughly that there are salts and minerals. They'll not ask you what mineral is present, like that they'll not ask you. There are other important topics to ask. Okay, so don't worry about that. Apart from the ABO blood grouping system, we also have something called as the RH factor. Now this RH factor, if it is there, it is positive. If it is not there, it is negative. 80% people have positive only. If you do not have that RH thing, then it is negative. Usually we don't have an antibody that is automatically present, but in case for some reason that blood comes in contact with the RH factor, only then the antibodies will be developed. So when the mother is negative but baby is positive, the baby's blood will come in contact with the mother and it can cause the antibodies to be generated. So first pregnancy, there will not be a problem. But second pregnancy, when it happens, what happens is the mother's body's antibodies will attack the baby. Death is a extreme case, but lot of children can be born with severe anemia, jaundice and other things at birth. So this is called as spelling mistake. 1 second. So this is called as erythroblastosis fetalis. Okay. This is the condition wherein the positive blood group of the baby can trigger antibodies in the negative blood group mother. Clear, everybody? Yes, is that clear? Good, RJ, good, very good. Okay. Now, when you look at the WBCs, WBCs can be agranulocytes or they can be granulocytes. Okay. Now, when you say granulocytes, they have some tiny granules in their structure, and that includes the neutrophils, the basophils, and the eosinophils. Okay. Now, neutrophils, they are the first ones to come when there is a problem. Basophils are involved in inflammation and eosinophils are involved in allergy. Remember that. Okay. Now, agranulocytes comprise of monocytes, okay, as well as lymphocytes. Clear, everyone? That's it. Okay. And when you look at the platelets, right? Now, these platelets are involved in coagulation of the blood, I said, correct? Now, whenever there is an injury, what happens? All the platelets will come and they will start releasing some factors. Okay. So, the platelets, okay, platelets are going to release some factors, and these factors will go and wake up someone known as the fibrinogen. So, this fibrinogen will be converted into something called as fibrin. How does that happen? They directly don't wake up someone called as a fibrinogen. Instead, what they do is they go and wake up someone called as the thrombokinase. He is the enzyme that converts someone called as prothrombin into thrombin. That's it. So, once the thrombin is ready, thrombin will go and convert the fibrinogen to fibrin. Okay. So, actually speaking, fibrins are not directly present in the plasma. It is the fibrinogens that are present. Okay. Clear, everybody? No, RJ, don't worry. That level of this thing and all, they'll not ask you. And just so you know, they're not completely the same. Okay, the nucleus is slightly different. Okay, if you look at neutrophils, they have a multi-lobed nucleus, whereas a basophil has a single-lobed nucleus. So like that, they have some differences. But nevertheless, when it comes to your exam, they will not ask you that kind and all. Okay, and given that when you're printing the question paper and all, it's very difficult to print that much of accuracy. So don't worry, those kind of diagrams may not come. Okay, is that clear? But for those of you who are attempting next year for the CBT mode, might be possible, but there are a lot of other important things, so focus there in your preparation. Clear, everybody? Now, when you look at lymph, okay, lymph, if you see, is the major fluid that is released into the extracellular space. Now, what happens is all my blood vessels, my arteries are going to divide into smaller arterioles. These arterioles again branch into fine capillaries and they reach my tissue. Now, in the capillaries, they are so small and so tiny and so fragile that sometimes the blood will ooze out. A little portion of the plasma will ooze out of these vessels, forming the lymph, which is nothing but the tissue fluid, extracellular tissue fluid. Okay. Now, this fluid, when released into the extracellular space, enters the lymphatic vessels. Now, these guys form a whole channel. They have lymph nodes, we have lymph organs, and ultimately they are returned back into the heart. Okay, but in this process, okay, now along with the plasma, a lot of WBCs, a lot of antibodies, immunoglobulins, all of these are there in my lymph. So for defense and for exposure to the antigens, all my cells need to come to the lymph organs only. Now, if you look at the lymphoid organs that are present in our body, okay, firstly, we have primary ones. Okay, so we have primary ones that include your bone marrow as well as your T lymphocytes. Thymus. Okay, so the ones that mature and proliferate and grow in the bone marrow are called the B cells, and those that do the same in the thymus are called as the T cells. But where are they produced? All of them are produced in the bone marrow itself. Okay, is that clear to you? Now, when you look at the secondary lymphoid organs, now they grew up, they got trained, okay, but where do they go and fight? Now, for example, all of your schools have something called as your NCC, right? So if you are a part of NCC, you are made into a soldier, you're trained, you are given all the exposure, but not like you're actually meeting the actual war ground, right? Now, once you are ready and once all the proliferation is done and once you know how to differentiate your cells and other cells and everything is complete, training is complete, you will be sent to the border to actually fight. So that is the secondary lymphoid organ. So that includes your Peyer's patches, your tonsils, your lymph nodes and everything. And this is very, very important because this is where the actual antigen exposure is majorly happening in the body. Okay, everyone, is that clear? Yes, that's great, RJ. I hope you'll not forget it. Okay, clear, everybody? That's about it. Okay, so lymphoid organs is super important. Also remember there's something called as MALT, okay, which is nothing but mucosa associated lymphoid tissue. So wherever we have a problem like a mucosa layer, we'll have little bit of lymphoid tissue there also, because whenever there is a problem, whenever somebody is captured, immediately I can go and attack and kill them. So I have something called MALT, but I'll explain all of this to you again when we're doing human health and disease, so that time it will be even simple to you. Okay, everyone, is it clear? Fantastic. Okay, now coming to the heart. Okay, now this image does not do justice because sometime later I'll show you the actual app, or let me see if I can share my access so that you can see how the heart actually is beating. Okay, and it's one of the most fascinating things for us to watch. Now, if you see, the heart is mesodermally derived and it has 4 chambers in the mammalian heart. And if you see the wrapping paper around it, the protection cover around it, we call this as the pericardium. Okay, and now this pericardium again is double walled and there is a small fluid that is present, which is the pericardial fluid. Again, it is going to function for shock absorption, for lubrication, making sure that the heart is not damaged in any way. Okay, clear to you? So inside this pericardium we have the heart. Now heart has 4 chambers, right? Yes, RJ, I'll answer your question 1 minute. Okay, so the 4 chambers that we see, so usually for remembering purpose, I always make the Dabba diagram. Okay, so in the Dabba diagram, if you see, we have a right auricle, left auricle, right ventricle and left ventricle. Okay, 4 chambers of the heart we have, and between the 2 auricles we have an interauricular septa. In between 2 ventricles we have an interventricular septa. Okay, now these ones in between the auricles and the ventricles we call as the auriculoventricular septa. Okay, is that clear, everybody? Perfect. Okay, now let me answer some questions. Okay, so RJ, what happens is, see, the capillaries are so thin, no? So like the diffusion is happening in the alveoli of your lungs, same way here also, in the pressure that the blood is going, little bit of fluid will go off. Okay, you cannot stop that. Okay, that is how it is going. You don't have to know very much details. Okay, there is a lot of processes that happens and all that. You don't have to understand all that. Just remember that these lymph and certain particles, just because of the pressure, no, they just quickly come out and then they'll join the system back again. Understood? Okay. Yes, RJ, definitely auricular septa will be thinner than the ventricular septa. Okay. Yes, RJ, I did. But see, the thing is, we don't have the time for me to show you the entire dissection, which I will maybe sometime down the year, but not before your exam. Okay, I don't want you all to get fascinated and get distracted. So let's focus back on the chapter and let's make sure that we are keeping all of this very, very clear in our minds. Okay, clear, everybody? Perfect. Okay, so in these 4 chambers that we have. Okay, what we need is a few cusps in between. Okay, so they have a cusp in between. Okay, I'm so sorry, am I clear now? Am I audible? Right. Okay, so the 4 chambers that we have, let me clearly make this thing for you. So we have a right auricle, left auricle, a right ventricle and left ventricle. Okay, now between the right auricle and right ventricle, we have some valves. Okay, so we have a valve. Now if you see, if you write 1, 2, 3, 3 comes on what side? This is the right side. So 3 cusps are present on the right side and 2 is coming on which side? The left side. So that is coming on the left side. Okay, so bicuspid and tricuspid valves are present. Why do I need these valves? When I'm squeezing my ventricles and auricles, I don't want backflow of the blood to happen. So that is why I'm going to restrict the blood to the respective chambers and make sure that the blood is going in only one direction. Okay, so for that I'm using these valves. Now same way, once I finish squeezing out all the blood from my ventricles, what happens? The blood should not come back inside my ventricles once I'm done. Correct, during the process. So in order to prevent from that, I have some semi-lunar valves that cover both my ventricles. Okay, now from the left ventricle, I have the aorta. This is the major blood vessel that is carrying the oxygenated blood all the way to the body tissues. Okay, and this is bringing the deoxygenated blood back to the heart into the right auricle. Now who is bringing this? It is through the vena cava. Okay, from the upper parts of the body, the head and the forearms, we have the superior vena cava, and from the thorax, the abdomen and the lower limbs, we have the inferior vena cava. So end of the day, both my vessels will fuse, forming one main branch of vena cava, attaching itself to the right auricle. Now who is this blood? This is the deoxygenated blood. Now this deoxygenated blood will come down directly into my right ventricle. Now from here, I have a very important blood vessel. Okay, I have a very important blood vessel called as the pulmonary artery. This pulmonary artery means away from the heart. Simple as that. Okay, so away from the heart is artery. Now this pulmonary artery is carrying the deoxygenated blood, and where will it go? It will go directly to the lungs. Okay, and there it is going to get oxygenated. Okay, so all this carbon dioxide is given to the lungs, all the oxygen is taken. This diffusion is what we spoke about in the previous chapter. Done? Now once this is completed, I am going to bring so this is deoxygenated, this is deoxygenated. Once it goes through the lung, this has become oxygenated. I bring the oxygenated blood back into the left auricle through what we call as the pulmonary vein. Okay, pulmonary because it is going through the lungs. Simple as that. Okay, now once the oxygenated blood has come, simply it is dropped down into the ventricles and the aorta is going to pick it up. Okay, is that clear? So simply one side of my vessels, one side of the circulation is taking the oxygenated blood, going to all the body tissues and then bringing it back into the heart. Correct? This is what I call as the systemic circulation. Why? Because it is going through all my systems. No, that is why it is called as my systemic circulation. And then on the other side, it's going through my lungs. So I will call it as the pulmonary circulation. Okay, both together we call as the double circulation. That's it. Done. Okay, yes, Arun Kumar. We are going to do entire genetics tomorrow morning. Okay, so just be ready for that. Okay, Perfect. So I'll do all 3 chapters, principles of inheritance, molecular basis and evolution tomorrow morning. So definitely come back to class tomorrow morning. Don't forget. Okay, everybody? Perfect. Okay. So double circulation, I hope it is clear to all of you. Now I'm going to teach you something called as the cardiac cycle. Okay. Now what is cardiac cycle? Nothing but the squeezing and the relaxing of the chambers. Okay, the squeezing, extra pressured one we call as the systole, the relaxation we call as the diastole. Okay, so contraction and relaxation is going to take place here. Okay, now this contraction and relaxation is definitely controlled by some signals which I will tell you at the conduction part. Okay, now when we are looking at the cardiac cycle, first we start with a atrial systole. Okay, so the auricles are going to contract. Okay, so atrial systole is going to happen. Now once the atrial systole happens, what happens? All the blood is going to rush into my ventricles, correct? Now once this is completed, now my ventricles will start contracting. Ventricular systole will happen. Now, when the ventricles are contracting, what's going to happen? My blood should not get packed into my auricles. So, in order for that to happen, I am going to close these valves. Okay, my bicuspid and my tricuspid valves are going to close, causing a lub sound to come. Right after my ventricular systole, to stop the backflow, my cuspid valves are going to close, forming the lub sound. Clear? Now once this is completed, okay, it will come down. Once the blood has come down, in the process of squeezing, what will happen? It will enter the respective blood vessels. Now once it has entered, it has to relax. So now a diastole will happen. So once this contraction was over, this guy went to relax. So atrial diastole happened. Now ventricular systole, once it's complete, now ventricle also will enter the diastole. So this is called as the joint diastole. Okay. Now once the joint diastole has happened, what happens? Like I said, during the relaxation, the blood should not come up as well. So my semilunar valves are shut. So this is what causes the dub sound. So this lub and dub is nothing but the sounds of the valves closing. Clear, everyone? Is that clear to all of you? Yes. So this one entire cycle takes about 0.8 seconds, and that's what we call as one cardiac cycle. Okay. Now the amount of blood that comes out after every cycle is what we call as the stroke volume. Okay, which is about 70 ml roughly. Okay, now if this is 0.8 seconds, means per minute I will have about how many cycles? About 70 to 72 cycles. Okay, so roughly 70 to 72 times my heart is going to pump in every minute, and every time the heart pumps, about 70 ml of blood is pumped out of the body. Okay, so that means the total amount of blood, okay, that my heart will pump per minute is what I call as the cardiac output. Okay, so this is per minute. So that is nothing but stroke volume into the beats. So stroke volume is 70 into 70 or 72. So roughly 7 to 7 is 49. So 4,900 is roughly 5 liters of blood is pumped out of the body. Now you can get very simple numericals asking you, okay, this guy's heart beats 100 times and every time the heart beats, stroke volume is 50. So what is the cardiac output? Simple, 100 into 50, 5,000 ml. That's all. Okay. So roughly 70, 72, average they say 72. So it can be 70, it can be 75. So it is just an average range. Okay, it can never be on spot for any person. Don't worry. Okay, is that clear? Now coming to the conduction. I hope this diagram is useful, because very easily you can see how the jumping of the conduction is going to happen. Okay, but I'll also show you with my double diagram. You can omit this image if it is confusing, but this is just the real image of a heart. I thought it will be fascinating for you all to see, and that is why I brought this across. Okay, so if you see, this is my right auricle. On the top corner, I have the SAN. Sino-arterial node. This guy is going to start my signals, because the heart does not listen to the brain. Even if the brain is dead, the heart can keep pumping. Correct? So the sino-arterial node is the pacemaker of the heart, which will start the signals. Now once one guy starts the signal, everybody will capture it and they will contract together as one unit. Okay. Now while this contraction is happening, this will jump to the AV node, and then through the AV bundle it will come down. Now this AV bundle is also called as bundle of His. Remember, okay. Now once it comes down, it will branch into the AV branches, the right and left branch, and from there it attaches to the different parts of the ventricles and it will supply the signal. So once the signal reaches, all the ventricles will start to contract. Okay, so by the time this jumps and comes to all the ventricles, the auricles has finished their systole. Understood? So this is how the conduction is taking place in the heart. Now these electrical signals we can monitor from the outside, and that's what we called as an electrocardiogram, which is ECG. So the graph we get is the electrocardiograph. So the graph predominantly has these peaks. So first we have a P wave, and this is nothing but atrial systole. Okay. Then we have a QRS complex. This QRS complex is nothing but the ventricular systole. Now in the background, as the ventricles are contracting, atriums are relaxing. Okay, but that won't show in the graph because it's very, very small comparatively. Okay. Now after this we have a T wave, which is nothing but the ventricular diastole. Okay. More than diastole or systole, I think we should talk about the electrical signals. So here you can consider this to be atrial depolarization, okay, ventricular depolarization and ventricular repolarization. Now where is auricles undergoing repolarization? When the ventricles are depolarized, this is background may happening all the repolarization, but that we can see in the graph because it's very, very small. Okay. Yes, absolutely, Disney crystals. So what's happening is the sun will generate about 72 signals every minute, and that's what corresponds to this entire cardiac cycle. Okay. Clear, everybody? That's all about your heart and everything. How the heart is regulated is definitely we have what we call as a myogenic heart. Okay, that means that it can generate its own signals. But from the nervous system, we definitely have the parasympathetic as well as the sympathetic nervous systems. Okay. So they will contribute to increasing the blood pressure, increasing the blood rates. We also have the adrenalines, okay, who can help us in increasing blood pressure, creating that rush. So all these hormones can regulate the heart very well, okay. And disorders of the heart includes firstly what we call as coronary artery disease, or also called as atherosclerosis. Okay, so this atherosclerosis is nothing but deposition of certain fat, iron, salts and all this in our capillaries, creating a block. Okay, so because there is a block, what happens? The blood flow is not very smooth. So over a period of time when the heart does not get enough oxygen, it gets tired and it'll be like, go bro, I'm going to stop. So when this happens, a person will get into a heart attack, which is nothing but a myocardial infarction. Okay. Now a very common disorder is blood pressure. Okay, or blood pressure is the wrong thing to say, because blood pressure is nothing but the pressure that we see in the blood vessels, which is usually 120 to 80. Systole to diastole, okay. But when you have hypertension, what does it mean? It means it is more than this value. something more than 140/90, okay? But all your body, all your other organs are basically designed for 120/80. So when you're under hypertension, definitely your other vital organs may be significantly impacted. Okay, so you've got to take care of yourself. All right. Now, apart from that, cardiac arrest is when the heart will stop completely. Okay, but at the same time, if there is inefficiency in the pumping and it is not pumping properly, you call it as a heart failure. Okay, so cardiac failure is different from a cardiac arrest. Apart from that, we also have angina. Okay, angina pectoris is the chest pain. If enough oxygen is not transported to the heart, you can have a significant pain. Okay, is that clear? Okay, so Vanavel, just for everyone to understand, I hope that's okay with you. Okay, fantastic. Okay, beautiful. So the last one I said was angina. Okay, so angina pectoris is the chest pain because of not enough oxygen to the heart. Okay, that's it. We're done and we're going to start with excretory products and their elimination, guys. So today at a stretch, we're going to finish all the human physiology chapters. Do subscribe, and tomorrow morning we're going to do the entire genetics unit. Okay, so stay tuned, subscribe, and definitely do join into the class tomorrow morning. Is that clear, everybody? Okay. Perfect. Okay. Now, coming to excretory products and their elimination. First thing is you need to understand the human excretory system, how urine is formed, the function of the tubules, countercurrent mechanism. All this will come in the process of forming urine itself. How the kidney is regulated, the role of other organs, and disorders. I think you can't see that part. So, disorders is the last thing that you will have to study. Okay. Is that clear, everyone? Clear, everybody? Perfect. Okay, let's start with excretory products. So when you look at excretory products, either this product can be ammonia, which is the most dangerous, and we call these organisms as ammonotelic. Okay, so aquatic insects, okay, cartilaginous fishes, okay, and yeah, anybody in the water basically has a lot of access to water. So immediate diffusion, say, you can chuck this ammonia out. Okay. Now, when you look at urea, it is moderate toxicity. It's not like you can't store. It's not like you need too much water also. Everything is moderate. And usually mammals, okay, and a lot of terrestrial forms will follow this particular mode of excretion, and we call these organisms as ureotelic. Okay, now when you look at the last type, that is uric acid. Now when you don't have access to water at all, okay, usually terrestrial reptiles, okay, birds, they all need to store this for a longer period of time in their body, and that's why it uses very minimal water. Okay, so hardly any water. So that means it is given out as in the form of pellets or paste. Okay, it's not going to utilize any water and is non-toxic also, so you can store it in the body for a longer period of time. Okay, now coming to the human excretory system, very simple. We have a pair of kidneys. Okay, and they are present just at the intersection of the thoracic and the lumbar vertebrae. Okay, and they are guarded by the last rib of the rib cage, and also they are going to continue into the abdomen. Okay, now from the kidney to collect the urine down into the bladder, we have the ureter. Okay, and the urinary bladder is what stores the urine until we are throwing it outside the body. Okay, and then at last we have a urethra, and this urethra is the tube through which we are going to take the urine from the urinary bladder to the outside. Okay, and it opens with a urethral meatus. That's the opening of the urethra. Is that clear to you? Now, if you look inside the kidney, now kidney you already know it's a reddish bean-sized organ and all that. But inside the kidney, now first of all, every organ has a wrapping paper, I have told you. Now, this wrapping paper here is called as the renal capsule. That's all. Okay, it's a huge envelope that is covering the kidney. Now, inside the kidney, we have two distinct places. One is the outermost cortex and another is the innermost medulla. Okay, now inside we have this space which we call as the renal pelvis. Okay, and this pelvis is where all the urine will come and fall. The ureter is going to pick up the urine from the pelvis and bring it outside. Now for the ureter, for all the blood vessels to enter and exit, we have a notch here called as the hilum. Clear? Is that clear to all of you? Now, this cortex tissue, like you can see, is penetrating into the medulla in between these pyramids, correct? Now, these structures are called as the columns of Bertini. Okay, and these triangular structures, right? In your textbook, you might have seen the exact triangular picture of the diagram, and that is what you call as the medullary pyramid. Okay, is that clear? Now I'm showing you this particular image. You might think it's not your NCERT, but let me tell you that this is basically just for you to understand and keep that image in your head. So whenever you see the diagram in your NCERT, you will associate to this image and you'll be like, ah, yeah, ma'am showed us, no, so we will not forget it. Okay, that is why I'm showing you these images. These are real simulated images and these are not your exact NCERT diagrams, but you will find all of these portions there. So just for helping in your retention and for helping in your memory, I'm showing you these images. Is that clear to all of you? Everybody? Perfect. Now, if you look inside the kidney, like I said, we have a cortex and medulla, but in that we have special cells or functional and structural cells known as the nephrons. Now, if the nephrons stay in the cortex area itself, then we call them as cortical nephrons. Okay, but if they come deep inside like this one, see how deep they are going inside the medulla, we call these as the juxtamedullary nephrons. Okay, is that clear to all of you? Now going into who actually these nephrons are. Let me tell you, these are the single cells or the units that are single structures that make up the kidney, and these are the ones that exhibit all the functions that you need for the kidney. Okay, so what we have here first, okay, we have the blood vessels bringing in the blood that have a lot of urea, that have a lot of nitrogenous waste, all unwanted materials are coming. Okay, now they will form a mesh-like system. Okay, and they will exit as a cleaner blood, and this is the efferent arteriole. Oops. Yeah, this is the efferent. Why is it disappearing? That's funny. Okay, this guy is disappearing. Let me write it somewhere here. So that is the efferent arteriole, and who is coming in is the afferent. So A comes first, E for exit. Always remember that. Okay. Now further, we're going to have a cup-like structure in which this blood vessel is present. This cup-like structure is what we call as the Bowman's capsule. Okay. Now from this Bowman's capsule, we have the first convoluted area. Convolutions means this ups and downs of the tubules. Okay, now this we call as the PCT or the proximal convoluted tubule. Now from there we have a whole loop of Henle that is going to dip in like a hairpin bend. Okay, this is called as the loop of Henle. Now after this we have another small convolution that is taking place, which is far away from the first glomerulus and the Bowman's capsule that we spoke about, and that is what we call as a DCT, distal convoluted tubule, and this will give rise to the collecting duct. So this collecting duct, not only from one nephron, but it will collect from so many different nephrons and take it down. So accumulation of all the DCT, of all the collecting tubules is what we see in the medullary pyramid like tubules. Okay, and all of them will drop the urine into the pelvis. Understood? Is that clear to all of you? Yes, everybody, don't lose your energy, guys. We're almost halfway through. Okay, we're going to make this happen. Just another 1 hour maybe, and we're going to finish off the entire human physiology. Very, very simple, easy concepts these are. Okay, the moment you understand the concepts and you understand how it functions and everything, then it's going to be super easy for you to apply it in your exam as well. Understood? Clear, everybody? Perfect. So now if you see these nephrons, the structure you've understood, right? Now the PCT, what it does, the loop of Henle, what it does, the DCT, what it does, is all that comes under the functions of your tubules. Now, as you understand the function, you can also understand how the urine formation is going to take place, and that's exactly what I'm going to explain to you here. Okay, so first understand that over here the glomerulus is going to put so much of pressure, okay, to the blood that everything is going to get filtrated and come out. So this process is what we call as ultrafiltration. Through ultrafiltration, I'm going to filter everything I have. I have some tiny podocytes also that are present in my Bowman's capsule, slit-like structures. So that also will filter out and everything will come inside. Okay. Now, once it comes inside, I'm not going to filter who is there, who is not there and all. Everybody, I'm just going to like train them down, juice them down and bring them in. Okay. Now, in this process, there must be water, lot of elements that I want. Correct? So that will be reabsorbed in the PCT. Okay. So if reabsorption has to take place in the PCT, okay, that means that this particular PCT must have extreme surface area. So that is why we have a brush border appearance and we have cuboidal epithelium here to help us with the reabsorption. Okay. So 99% of the filtrate is going to be absorbed back. Now, what is there in that 99% filtrate? 70 to 80% is only water. Apart from that, all the ions that I want, minerals that I want, everything I'm going to reabsorb back in my PCT. Okay. Now, when I'm dipping down into the loop of Henle, there is a very important function that happens which causes the urine to concentrate. Okay, is that clear? So, in the process of this concentration, what I'm trying to do is, nobody should be affected around me as well, correct? So, in order to maintain that, I'll tell you. What happens is, this particular efferent arteriole, instead of going home from there, he goes like this, covers the entire nephron once and only then goes. So, all these capillaries are called as peritubular capillaries. Okay, and what we have here around the loop of Henle is what we call as the vasa recta. Okay, now vasa recta is going down like this and coming up like this. Now, if you look at the loop of Henle, this is my descending limb, this is my ascending limb. Okay, is that clear, everybody? Okay. So now what is typically going to happen is, when this loop of Henle is going to bring the filtrate down, remember a small trick, okay? One second. Let me tell you the logic and then it will be easier for you to understand. So see, if you look at the alphabets, A comes first, D comes next, correct? A, B, C, D. Then if you say the alphabets till the end, you will see that T, U, V, W. So S comes first, then W comes. Correct? So same logic here also. Ascending limb, this one of the loop of Henle, is permeable to salt. This particular descending limb is permeable only to water. So what happens? This guy, when he is coming down, he'll give out water. Okay? So he'll give out water, and from 300 milliosmolar, he'll get concentrated and reach 1200 milliosmolar. Means he is getting almost 4 times more concentrated. Okay, is that clear? Everyone, Sendil Kumaran, don't worry. Let the re-NEET finish first. Let's focus on re-NEET first. Okay, don't worry about that. Okay, so coming to what the loop of Henle does. it will give out the water. So urea, the salts, nitrogenous wastes are going to get more and more concentrated. Now when it is going up, what happens? Here my ascending limb is permeable to what? It is permeable to the salt. So here I'm not going to give out urea. I'm going to give out NaCl. So when I go up, I come to a much more concentrated urine where I've given out water, I've given out some salts, and I've become 4 times more concentrated. But why this is happening? Understand that this is happening because one side I am giving out water here and I am picking up and I am giving out NaCl here. But why should I get concentrated? If this interstitium, medullary interstitium is not maintaining this concentration, I typically cannot do this concentration process. So who is helping me? This vasa recta, when it is going down, it's going to pick up this NaCl inside the vasa recta, come down, maintain this gradient of concentration, and while it is going up, because lot of salt, water is there, it will give out the salt here. Because lot of salt is there, it has too much of it has lot of water outside, too much of salt is there inside, so gives out the salt into the water and it goes back. So when the vasa recta is leaving, it leaves with a 300 milliosmolal that it came with. So there is no difference in the blood. But in the process of dipping, what it is doing? It is helping me maintain this concentration in such a way that I can definitely carry out my concentration without any problems. So when the filtrate reaches the DCT, it is concentrated. Now this mechanism is what we call as the counter current mechanism. Because this fellow is going like this, that fellow is coming like this. Again, this fellow is coming like this, that fellow is going like this. So there is a counter current that is established. Okay, is that clear to you? Then we have the DCT. Okay, now what this DCT is going to do is, ultrafiltration is over, reabsorption is over, concentration is over. He's going to go out now. But in case I need to correct something, any conditional reabsorption of sodium or any secretion of bicarbonates, hydrogen, potassium, anything if I have to secrete, then I have a tubular secretion that is taking place here. Okay, in the DCT. So any correction, anything whatsoever, because PCT is taking up so much, he doesn't have time to select, he doesn't have time to listen to your instruction. But DCT has all the world's time. So he is going to slowly take up all this thing and he is going to give it off to the collecting tubule. Okay, now this collecting tubule also, while it is going, it will give out little bit of urea here so as to maintain this 1200 milliosmolal concentration so that counter current mechanism can happen peacefully. Okay, that's all about the formation of urine, and I hope you've understood all the functions of the tubules as well. Is that clear to all of you? Everybody? Yes. So ultrafiltration followed by reabsorption, followed by tubular secretion. This is all you need to remember. Okay, everyone. Now talking about urine and mixturation. Urine is the final product. We secrete about 1.5 liters to 2 liters of urea every day. Urine every day, and roughly about 25 to 30 grams of urea every single day. Okay. Now for the mixturation process to happen, definitely neural control is very important. Okay. So here what's going to happen is, once my urinary bladder is full, I have some stretch receptors here. Okay, now they'll tell my brain that, okay bro, bladder is full, you need to excrete now. So the brain will then give us motor control and say, okay, it's time to urinate. And once the sphincter muscles in the urea, so there is an internal sphincter and there is an external sphincter. Now you can only voluntarily control the external sphincter. Internal sphincter you cannot control. Okay, so the internal and external sphincters will relax, and as a result of which micturition or urination will take place. Is that clear to all of you? Now talking about the regulation, okay, now also remember one thing, that I need to talk to you about the GFRs, right? Now GFR is nothing but glomerular filtration rate, okay? What is the amount of filtrate I'm getting after every, in a minute is what I call as a glomerular filtration rate, okay? Now this filtration rate is directly connected to the blood pressure. You need to remember that. Okay, now when you look at the regulation, first is the hypophyseal regulation, means whenever I am going to have some whatsoever it may be, some kind of a loss of fluids that's happening, some receptors will catch them and tell my brain that, bro, you're losing too much of fluid. Now this loss is known as diuresis. Okay, now this loss is known as diuresis. So if this loss is happening, my brain will tell my hypothalamus to go and control this. So what my hypothalamus does, it will tell the pituitary to release something called as the ADH, the anti-diuretic hormone, to prevent the diuresis, or it's also called as the vasopressin. So once this is released, what happens is directly my reabsorption is triggered in the DCT, because DCT is the one who can understand the instructions. PCT, that does not have time. Okay, so now the DCT is going to continue reabsorbing. It will also go and call the other aldosterons from the adrenal gland, and as a result of which it will regulate, it will increase the blood pressure, it will increase the GFR, and it will increase the reabsorption in the kidneys. Okay, now renin-angiotensin mechanism is where the DCT, okay, while it is going back home after forming all the vasa recta and everything, it will come in contact with the afferent arteriole. Okay, now the distal convoluted tubule, not the efferent arteriole, the afferent arteriole along with the DCT, when they meet each other, they have some sensitive cells produced. These sensitive cells are called the juxtaglomerular apparatus, and they release something called as renin. Okay, now once the renin is released, what happens is this renin will go and wake up someone called as angiotensinogen. This angiotensinogen gets converted to angiotensin 1 and then angiotensin 2. This angiotensin 2 is a vasoconstrictor. So BP increased, GFR increased, and as a result of which reabsorption is also increased. Now both of these mechanisms will increase the blood pressure, increase the GFR. If they are doing too much and the heart can't bear it, the heart is going to release something called as the atrial natriuretic factor. Okay, now once this factor is released, the blood pressure is brought down, because vasodilation will happen. Okay, it will bring down the blood pressure, it will bring down the GFR as well. Clear? In all of these processes, you can regulate your excretory product and the regulation of the excretory system. Okay, now coming to the disorders, guys. This is the last part of your excretory product chapter. Now for your disorders, first thing is glomerulonephritis. Okay, what is this? Inflammation of the glomerulus is taking place. Secondly, what is happening? Apart from the glomerulonephritis, we have the kidney stones, which we call as the renal calculi. Okay. Apart from that, what else do we have? We can have some, so renal calculi is nothing but stones. These are nothing but salts. Okay, they crystallize and they form in your kidney. Okay, apart from that, we can have extra glucose or extra ketone bodies in the blood, in the urine, which indicates conditions like diabetes mellitus. Okay, now in case the ADH hormone is not functioning, then what happens is it will create too much loss of fluids from the body because ADH is not working, and that is what we call as diabetes insipidus. So these two are the type 1 and the type 2. Okay, is that clear, everybody? Clear to all of you about the disorders? Now in worst cases, renal failure can happen, where the kidneys don't function anymore, okay, and they're like, I give up, bro, I can't do anything more for you, right? So when that kind of a situation comes through, okay, when that kind of a situation comes through, this is when so this is when the particular kidney cannot remove any more of nitrogenous wastes. Correct. So they cannot remove. Who will remove this? For that only we can do something called as dialysis, which is an artificial kidney. We can take out the blood from a blood vessel, send it through a dialyzer. So what it has fluid just like plasma without the nitrogenous waste. So when the blood will see this, it will be like, okay, there is no concentration, here there is high concentration, so through diffusion the nitrogenous wastes are removed, and then I can bring it back into the body and everything will function as normal. But the other processes that the kidney does for you, certain hormones that it releases and all, it cannot help you, but overall it can clear up the nitrogenous wastes without a kidney. So like an artificial kidney, it can help you. And in worst cases, you can transplant, but again, tissue matching, all that is very, very important. Okay? Is that clear to all of you? Everyone? Any questions so far? Any questions, all of you? Any doubts that you all want to clear? Nothing? Can we move on to the next chapter? Right. So here we are at locomotion and movement. Okay, everybody? Yes, just throw some stars and create some energy, guys. We're already halfway through. Okay, so when you look at locomotion and movement, you have to understand different types of movements, muscles, skeletal system, joints, and disorders. That's about it. Okay, so when you look at the types of movement, you have amoeboid movement, okay, which primitive organisms like amoeba and all that will exhibit. Okay, apart from that, you have flagellar movement. Okay, flagellar movement. So flagellar movement is with the usage of flagella. Okay, so this flagella will move and it will cause the movement. Okay, now same way we have cilia. Okay, and we have other microtubular structures which can help in our major transports, movements. Okay, now muscular movement is the most advanced type of movement, and this is both voluntary and involuntary, and it uses certain specific tissues, okay, which we can call, and these specific tissues are basically going to be the muscles that exhibit this entire movement. Clear, everyone? So when you look at muscles, okay, simple, we can classify them as striated or voluntary, or also called as skeletal muscles. Okay, these are involved in all the somatic movements of the body. And then you have a smooth muscles. These smooth muscles are unstriated and they are also involuntary. They are found in all your blood vessels, your internal intestines, all your internal organs. You will find this kind of a system. Okay, clear, everyone? And then when you look at the cardiac muscles, these are a different type of muscles altogether. So these cardiac muscles are definitely found only in the heart. They look like a skeletal muscle in the sense they are also having a striated appearance, okay, but nevertheless they are going to work tirelessly for you every single day. Okay, and now if you see, there is a significant branching that you can see in the case of cardiac muscles, and this branching is majorly because there is a need for all the muscles to contract and expand in a single unit. Okay, they all have to work together. They cannot do one thing at a time. So all the entire auricle should get the signals at the same time, ventricles should get the signals at the same time. That is why cardiac muscles are having these intercalated discs to connect the muscle fibers. Okay, is that clear? Now coming to the ultrastructure of a muscle. Okay, if you look at a muscle, first of all, inside the muscle you have certain certain fibers, okay, muscle fibers. Now what are they made up of? What is the myofibril made up of? We have first of all a molecule known as actin. Okay, now this actin molecule has a component called a G-actin, which is the globular form, which forms a filamentous actin. Okay, now this filamentous actin is like 2 strands, okay, and in between we have one line, one strand called as the tropomyosin. Okay, so this is the actin, this is the tropomyosin, and inside here and there we have some subunits which we call as the troponin. Okay, together these will form the actin filaments. Okay, now from this actin filament, okay, we have a thin band that is formed. Okay, so because it is very thin, we call it as an I band. I, very thin. Okay, I letter is only very thin. No, that is why I band is also very, very thin. Remember that. Okay, the other molecule that you have is the myosin molecule. Okay, so what the myosin molecule has, it has a light part and a heavy part. Okay, so the light meromyosin is there and then the heavy meromyosin is there. So the head region has two spots. Okay, one is for ATP binding and for ATP to get hydrolyzed. The other spot is for binding to the actin binding site. So here also there is an actin binding site, but unfortunately this troponin is covering that actin myosin binding site. Okay, understood? Is that clear? Guys, come on, throw some stars and fire in the chat box, man. I need energy. That's the spirit. Okay, so these two molecules together are forming the major filaments of my ultrastructure. Okay, so this myosin is going to form the thick band, and thick means it is very fat. So that is why it is very broad, no? So we call as the A band. It's not called because it is thin or thick, okay, but I am helping you remember it because most students get confused between a I band and an A band. Okay, is that clear to all of you, everyone? All right. Okay, so if you see here, right, if you see here, these actin bands form the thin bands like this, okay, and my myosin bands form the thick bands like this. Now the middle of this myosin band I call as an M line, okay, and this zone where I have both of them separated and I can see the myosin is my H zone. Okay, now this is the I band, this is the A band. Okay, now between the I band, I have a Z line. Okay, so between two Z lines, I call this as my sarcomere, and this is the structural and functional unit of my muscle. Clear? Now, how exactly this contraction of a muscle is taking place is through a sliding filament theory. Okay, very simple. I'll teach you what a sliding filament theory is. Okay, so first is my CNS is giving a signal, okay, to the neuromuscular junction. Okay, so once the signal is received, what happens is the sarcoplasmic reticulum will release the calcium ions. Okay, now this calcium ion will go to the troponin molecule, and once the calcium is bound, okay, it will unlock the myosin binding site. Okay, so once the site is unmasked and it is open now, what happens? ATP is hydrolyzed, okay, and the myosin will come and attach, forming a cross bridge. Okay, so what is happening? My myosin molecule is taking the ATP, okay, it's hydrolyzing and going and attaching to the I band. This is my A band. Okay, now after attaching, this guy is going to pull it inward in such a way that the I bands will be brought inside and they will slide on top of each other. So as a result of which, what will happen? I band will decrease because they are sliding, no. So they are sliding, the total bandwidth it's going to reduce. But they will slide as long as they reach the A band's length. So the A band will not change. It will remain unchanged. Remember this, very, very common NEET question this is. Okay. So here my A band is getting is remaining unchanged. My I band is decreasing. My head zone will disappear. Okay, and my entire contraction will take place. Now once this is done, once the ATP is hydrolyzed, the cross bridge is broken. Okay, now once the cross bridge breaks, again if there is a signal and ATP is hydrolyzed and again cross bridge formation happens, there will be a contraction that takes place. Is that clear to all of you? So as a result of which, what happens here? Clearly, I'm going to have a very, very clear setup of sliding in. Okay, cross bridge formation is happening, contraction is happening. If no signal is there, then it will break. Okay, so once there is no signal, the calcium ions will go back into my sarcoplasmic reticulum. Everybody will sit inside. No questions asked. No site is open for cross bridge. So there is no formation of this thing at all. Okay. Is that clear to all of you? Any questions in this? Is it clear, everybody? That's all about your muscular system. Now coming to the skeletal system, everyone. Skeletal system made up of bones. You know that a few places we have cartilages like the ear pinna or the tip of the nose. But apart from that, the bones are the ones that give us the framework. They bear our weight and they're basically doing everything for us. Okay, is that clear to you? So now what you need to remember here is the skeletal system has two components, which is the axial system and the appendicular system. Okay, now the axial system is nothing but what all you will see in the center. Okay, which is basically the skull, the vertebral column and the rib cage. This is your axial. Your appendicular skeleton is everything outside and the girdles that are helping you connect this outside to the center. That's it. Okay, so axial and appendicular together make up 206 bones. Axial has 80 bones and appendicular skeleton has 126 bones. We'll see each of them in detail. So if you see here, firstly the axial skeleton has the skull. Okay, skull has 14. Okay, before I go to the facial bones, let me talk about the skull in itself. The brain box, the cranium has 8 bones and all these 8 bones are fused. Okay, so they have no movement at all and they are completely fused. Then we have 14 facial bones. Clear? So that makes it 22 bones. Apart from that, the ear ossicle has 3 bones each. Correct? Malleus, incus, stapes. So 3 bones each means we have a 6 ear ossicles that are present. Additionally, 1 hyoid bone is there here. Clear to all of you? So 8 plus 14 plus 6 plus 1. All of this is coming under the skull in itself. Okay. Then when you look at the rib cage. Okay. If you look at the rib cage, you have 24 bones. Right? We have 12 pairs of ribs. Now the first 7 pair is 1, 2, 3, 4, 5, 6, 7 is directly connected to the sternum. Okay, so that is why 1 to 7 is called as the true ribs. Okay, whereas the 8th, 9th and 10th are only linked to the 7th, not to the sternum directly. That is why 8, 9, 10 is known as the false ribs. Okay, and the last two, like you can see here, 11 and 12 are floating ribs because they're only attached to the vertebral column behind. They're not connected to the sternum at all. Okay, is that clear, everybody? All right. Right. Okay, so I guess, right. Okay, so I guess there was a small connection issue probably. So I hope it is clear to all of you now, everyone. Yes, is it clear to all of you? Okay, I guess now it's perfectly all right. So let's continue, everybody. Beautiful. Okay, so when we look at the rib cage, we have 24 bones covered over here along with one sternum. That's coming part of the rib cage. Okay, then when you look at the vertebral column, we have 7 cervical vertebrae, 12 thoracic vertebrae, then 7, 12, 5 lumbar vertebrae, okay, and then 1 sacrum which has 5 bones fused, okay, and then we have 1 coccyx which has 4 bones fused. Okay, so together if you count all of these bones, okay, so here we have about 19, 20, 26 bones here in the vertebral column, 24 bones in the rib cage plus 1 sternum, so that makes it 25. Okay, so apart from that, 51, 52, 52 plus 6, 58, 58 plus 14 is nothing but 62, 72 plus 8, 80 bones present in the axial skeleton. Clear? No? Yes, makes sense? Everybody? That's it. Okay. Now, when you look at the appendicular skeleton, okay, if you look at the appendicular skeleton, first thing is we have girdles to attach the peripheral bones to the center. Correct. So, for that we have 1 pectoral girdle and 1 pelvic girdle. So, pelvic girdle is 1. We have a hip. It has 2 bones called the coxal bones. Okay. So, this coxal bone is actually a fusion of the ilium, ischium and the pubic bones. So, we have 3 bones making up the coxal bones, but they are all fused. Okay. Now, if you look at the pelvic pectoral bones, in the pectoral bones, you have 1 clavicle followed by 1 triangular bone called as the scapula. Okay, so clavicle is a curved bone that's connecting to the rib cage on one hand and then connecting to the bone on the other, whereas here the scapula is a triangular bone that is present behind the shoulder. Basically, this is the shoulder. The pectoral girdle is nothing but the shoulder. Okay, and this develops a process known as the glenoid cavity for my bone to come and attach. Here, the same cavity is produced called as the acetabulum. Okay, so we have 2 bones here, so 2 on each side, so that is 4 bones here. So 6 bones make up the girdles of the body. Okay, then we have the appendicular skeleton wherein every limb has 30 bones. So if you see, we have 1 long humerus, okay, then we have a radius and ulna, then we have 14 phalanges. Okay, we have a wrist which is basically carpals, and then we have the 5 metacarpals. Okay, so 1 humerus, right? Then radius and ulna is 2, radius plus ulna. Then we have 8 carpals, 5 metacarpals, 14 phalanges, that's basically 30 bones. Okay, so 30 into 2 is the forelimb. That's it. So 60 bones in your arms. Same setup is there for your legs as well. Okay, so you have 1 femur, then we have 1 fibia and tibula, then instead of 8 carpals, we have 7 tarsals, and 1 bone went here to be the patella. Okay, so 1 femur, then 2, which is the fibia and tibula, then we have 7 tarsals, okay, then we have 1 patella, then we have 5 metatarsals, which is basically what you see in the foot, and 14 phalanges. So that makes it 30 bones. So 30 into 2, 60 bones you will see in your feet. That's it. Okay, so 60 plus 60, 120 bones. In the girdles we have 6, 126, and then we have 80 bones in the axial skeleton, making it up to 206. Yes, Humaira, don't worry, they will not ask. They will not ask each of the facial bones or each of the skull bones will not be asked. Don't worry. You don't have those mentions in your book, so you do not have to worry about it. Okay? Now, when you talk about the joints, we have immovable joints. Okay? So, immovable joints are like your fibrous joints of your skull, where you cannot move them at all. Okay, so fibrous joints of the skull. Okay, now partially movable joints are there. Okay, partial movable. So, these partially movables are those that you see in your vertebral column. Okay, so the vertebral column has cartilaginous joints which are partially movable in nature. Okay, now the last one is freely movable. Okay, now those include your synovial joints. Now, who are these synovial joints? They have a small fluid called as the synovial fluid. Now, because of this fluid, you can happily move it as long as you want, and that includes your pivot joints like your neck, okay? Then ball and socket joint like your shoulder and hip bones, okay? Then you have saddle joints, you have hinge joints. So, all of these joints come under synovial, and all your fingers, your elbows, knees, everything is a synovial joint. So, it allows free movement in your body. Okay, and coming to the last part of the chapter, it is disorders. So when you look at the disorders of the skeletal and muscular system, so one thing is muscular dystrophy. This is a genetic disease where there is genetic degeneration of the muscles, progressive degeneration. As time passes, there will be loss of muscle movement, and there will be paralysis that can take place. The other one is myotonic. Okay. What do you say? So muscular dystrophy. Myotonic dystrophy. That is only muscular dystrophy. So myotonic dystrophy. The other one is myasthenia gravis. Now, students usually get confused between this myotonic and myasthenia gravis. So, remember that myotonic dystrophy means it is genetic. Myasthenia gravis is an autoimmune disorder. Okay, so this autoimmune disorder will go and attack the neuromuscular junction. So, whatever the brain is telling me, I cannot follow. I didn't even get the instructions, right? So, I cannot follow, and as a result of which, I do not have proper exhibition of my activities. Okay, now the last disorder you should know about is tetany, wherein if there is very low calcium in the body, what happens is there is a loss of calcium. So calcium is very important for my contraction. So if calcium is not there, then contraction will not happen, and as a result of which there is a lot of movement problem. Okay, so rapid spasms and wild spasms can happen because of tetany. Okay, now apart from that, osteoporosis is a very important disease wherein mostly older women experience this, where the bones become brittle and you're very prone to a lot of fractures. Now this majorly happens because of a drop of estrogen, okay, because estrogen is the one that's helping in bone health. In especially women who have undergone menopause, the bone health will reduce, and as a result of which it can lead to very, very brittle bones and fracture. Okay, so Humaira, don't worry, animal kingdom included, I will be taking all the chapters for you, so definitely do subscribe and stay tuned. Okay, so today itself we have revised 4 chapters of human physiology. We have only 2 more left, and trust me, compared to the last 4, this next 2 are super simple. Okay, and tomorrow morning we'll be doing genetics, so don't forget to subscribe and tune into the tomorrow morning's class as well. Okay, Arjun, yes Arjun, you will be having 2 acetabulums because we have 2 sockets for the bones to fit, right? So 2 coxal bones and 2 acetabulums will be there. Okay, clear? Any other questions? Shall we continue? So neural control, very simple chapter, guys. So when you look at the neural system, definitely this is a very close to my heart chapter, because I feel like the brain is such an interesting organ, and I think that is something that, you know, keeps us all curious, right? Because there is so much in the brain that we still don't know of. So that is why, you know, it's more and more fascinating to look at, right? Alan Entertainment, we've just got another 2 chapters to complete and then we'll be done. Okay, yeah. Superb. Okay, so definitely do subscribe, guys. So let's start with the neural system. Okay, so if you look at the neural system, we've seen a system that progresses through different phylums. So first we've had very simple ganglia coming together, then we had some, you know, small groups of cells called as a, you know, brain, and then slowly the complexity evolved, and we have reached to a very complex organ like the human brain. Okay, so the neural system in general works for the control of all the systems in the body. It is the master system, of course, and all the coordination also comes from here. Okay, all immediate actions, immediate things, whatever is there, everything will be coming across through the neural system itself. And those of you who feel like, ma'am, what about the hormonal system? Don't worry, hormones bring about a long-lasting effect. Okay, so nervous system is controlling everyday activities. Everything that you need to say, everything will be considered in the case of the neural system. Okay, Arjun, yes, like I mentioned, we have 2 coxal bones, no kanna. So in the coxal bones that is making up our hip, we have 2 coxal bones, so we have 2 acetabulum also. I hope that's clear to you. Yes, perfect. Okay, so guys, I just want to check. I hope I'm audible to all of you and I don't have any problems, hopefully. Are you all able to follow the class? Right? Okay, perfect. So now, along with control, coordination and everything, this guy is taking care of all the functions in the body, right? Every system works because the nervous system is collaborating with all of them, coordinating with all of them. Literally, okay? So the neural system is definitely super important, and when you look at the human nervous system, first thing is we have a CNS, central nervous system, and a PNS, a peripheral nervous system. So central nervous system comprises of the brain as well as the spinal cord. And when you look at the peripheral nervous system, we have different kinds of nerves that coordinate the different parts of the body to our central nervous system. Okay, is that clear to all of you? Now, when you're looking at the PNS, we can further divide it into the somatic, which will control all the voluntary movements, and the autonomic. That is going to automatically control every other involuntary action in the body. And when you look at autonomic also, further we can classify it as sympathetic, which will urge you for the fight or flight responses, and the parasympathetic, that is more focusing on the rest and digest mechanisms. Is that clear to all of you? Yes, so CNS is brain and spinal cord, PNS is basically somatic, autonomic, and under the autonomic you have the sympathetic and parasympathetic nervous system. That is what is your human nervous system. Is that clear to all of you? Now when you look at a neuron, this is basically the structural and functional unit of your body. Okay, so when you look at the structure of a neuron, firstly, like you can see here, the neurons are connected to the different tissues, the different receptors to collect the information through what we call as a dendrite. Okay, this dendrite will receive the information and we have a central cell body which is going to coordinate all the information, decide what we have to tell the next neuron, and this axon is going to pick up this signal and take it to the next neuron through the axon endings. Okay, this is the axon. Now, like you all can see, around the axon, we have special kind of cells called the Schwann cell. Okay, and this Schwann cell produces a specific kind of protein which forms what we call as a myelin sheath. Okay, now here there are places where you don't find this myelin sheath, correct, where the myelin sheath is missing. These places are called as the nodes of Ranvier. Okay, clear everyone? Is that clear to all of you? Everybody? Right, so dendrite, axon, dendrite, cell body and the axon. This is the fundamental structure. Now around the axon we have the Schwann cells and the myelin sheath. Wherever myelin sheath is missing, we have the nodes of Ranvier. Okay, now when you look at how these neurons are typically conducted and generated, before that I will just give you a glimpse of the different types of neurons that you can see. Okay, and then we will proceed with this thing. So if you look at the types, you definitely have to remember unipolar means only axon is there. Okay, and this is seen in the embryonic stages. Okay, then we have the bipolar. So bipolar means what? One axon and one dendrite. So this we can see in the retina or the auditory nerves and all that. You can find this particular bipolar. In the nasal epithelium and all, you can find the bipolar neurons. Okay, then multipolar is what I just showed you. In all the CNS, you can find the multipolar neurons only. Got it? Now, when you look at the generation of an impulse, okay, let me tell you that this axon membrane, okay, this axoplasm that's present inside the axon is negatively charged. Okay, lot of negative proteins are there. Along with that, I have potassium. Okay, now outside who is there? Lot of sodiums are there. But sodiums can't come inside that easily. Okay, now what will happen is whenever there is a signal, okay, whenever there is some kind of a stimulus, something, the light came on my face or I heard something, I wanted to turn something, some stimulus was there. Once that stimulus will come, what will happen? The stimulus will immediately open the channel. Okay, so stimulus will open the channel. Okay, now when the channel is open, what happens? The sodium is going to run inside. So once the sodium runs inside, what happens? First I was negative inside, positive outside. Because the sodium runs inside, my outside becomes negative, inside becomes positive. This is what I call as depolarization. Okay, and suddenly they're like, bro, what's happening? So this potassium now is like, yo, the sodium has come inside. Now I have to somehow chuck all of these people outside. I don't have a choice, right? So I somehow have to chuck this outside. So let me go and neutralize this. Okay, this potassium will go out. Okay, and this will cause the repolarization to take place. So as simple as that. Why depolarization is taking place? Depolarization is taking place because sodium is coming in. Repolarization is taking place because potassium is going out. Okay, so as a result of which we have this depolarization and repolarization. So once this part has become depolarized, it will pass on this to the next region. So then this part will get repolarized, this guy will get depolarized, again he will get repolarized, again this one will get repolarized. So like that, this depolarization wave will continue as long as it reaches the axon endings. Okay, now in the axon endings we have something known as the synaptic bulb. Okay, and we have a gap known as the synaptic cleft. Then we have the next neuron here. Okay, now this guy is there before the synapse, so he is called as the presynaptic neuron, whereas this guy is present after the synaptic cleft, so he is called as the postsynaptic neuron. That's it. Okay, clear to all of you? Now, what's going to happen is this synapse is nothing but the okay. Is my this thing a little clearer now? Are you all able to hear me? So sorry, I think I had a small network glitch. Everybody, are you all with me? Okay, perfect. Hemant, Raghav, Arjun, Sahanashri. Sahanashri, yes. Yes, Kanna, you're right. Okay. Now I can see. I was just wondering why my chats are not coming, and then I realized my connection. Okay. Yes, now I think the connection is better. I'm so sorry. There was a small glitch and hence I just had to disconnect. Right, now I think it will be clear. So what I was saying is we were talking about synapse, right? So I said a synapse, an electric synapse is like just connection. Okay, just moving on from this thing to another. That's all. It's just going to flow. Okay, but when you look at a chemical synapse, we have certain processes involved. Okay, so when you look at a chemical synapse, this is how it looks. Okay, so this is the presynaptic membrane. Okay, and presynaptic neuron, if I put it better that way. Okay, so the presynaptic neuron, once the signal is brought here, okay, the signal is going to induce the calcium channels to open. Okay, once the calcium channels open, what's going to happen is the vesicles present here contain certain chemicals known as neurotransmitters. Okay, so this neurotransmitters are going to be inside this vesicle, and once this calcium comes, these vesicles are going to come and dock onto the presynaptic membrane. Okay, and after docking, they will fuse with the membrane and release all my contents outside. So this is going to release all the neurotransmitters into my presynaptic, into my synaptic cleft, which is the gap. Okay, now once this gap has all my chemicals released, the postsynaptic membrane will have certain receptors. Okay, now these receptors will catch the neurotransmitters and they will transmit it into the postsynaptic neuron. That's how a chemical synapse is taking place. Okay, is that clear? Perfect. Okay. Okay. So don't worry guys, I know it is buffering, but I'm just trying my best. Okay, so we're almost done with this chapter. We only have one more left. Don't forget to join tomorrow morning's class. Okay, and I'll definitely make sure that all of these problems are sorted off by tomorrow morning. Okay, usually it doesn't happen, but I don't know why today there is a network this thing happening. So we'll just have that figured out. Okay, perfect. All right. Okay. Now coming to the central nervous system of the body. Okay, we have a brain, and this brain and a spinal cord together make up the central nervous system. Okay, now when we talk about the brain, yes, we have brain and spinal cord, but in your syllabus we only have the brain. The brain has a forebrain, midbrain and a hindbrain that is present. Okay, now if you see forebrain majorly comprises of the cerebrum and the diencephalon. Okay, now cerebrum is your center for memory, for processing, for thinking, okay, for all your intelligence, for any work you do, for your motor coordination, for your sensory coordination, anything you want, everything is going to come from your cerebrum only. Now if you look at the cerebrum, there are a lot of convoluted surfaces that are present. Correct. Now these convoluted surfaces increase the surface area and accommodate more number of neurons. Okay. Now when you look at the neuron arrangement in the cerebrum, what we have is all the cell bodies are facing outward and all the axons are facing inward. As a result of which there is a gray color that is established on the top because of the cell body, and a white color which is established inside because of the myelin sheath of the axons. Okay, now as a result, cerebrum looks grey. That's why cerebrum is also called as the grey matter. Okay, instead of asking do you have brains, you can ask do you have any grey matter. Okay, that makes you very, very brilliant. Anyway, coming to the diencephalon, but don't go and ask anybody if they have brains. Okay, it's none of your problem. But yes, coming to the diencephalon comprises of two parts, which is the thalamus and the hypothalamus. Okay, now thalamus is going to be the relay station. It coordinates different brain activities in the forebrain. Hypothalamus is the guy who regulates your hormones, okay, your feeling for hunger, thirst, okay, your body temperature. Everything comes from this guy. Okay, so typically if you see, this is the hypothalamus, and this guy on top is the thalamus. Okay, just for your reference. Okay, is that clear to all of you? Perfect. Now coming into the midbrain, we have four prominent structures called the corpora quadrigemina. Okay, and they are involved in auditory as well as visual reflexes. Okay, so these four, we have one which is going to be controlling visual, two controlling the auditory. Okay, so together we have four bulb-like structures making the corpora quadrigemina in the midbrain. Along with that, we have a small canal also that has a small fluid inside, and this connects the fluid-filled cavities of our brain. Okay, so forebrain is done, midbrain is done, and last we have the hindbrain, and the hindbrain contains the cerebellum. Okay, the cerebellum also has a very similar convoluted structure like the cerebrum itself, but here what the cerebellum does is, its function is mostly with regards to posture, balance, okay, and coordination of the body. Okay, and then we have the pons. Now, pons is the major relay station. Who and which signal should go to which part of the brain, everything will be controlled by the pons. Okay. Alan Entertainment, is it buffering again? Okay, I will just work on that. But yes, Raghav, definitely I will do a very small session that is for all the scientists. Okay, what they did, which year, everything I will culminate into a single session, and I will definitely bring it for you. Okay, don't worry, Raghav, definitely. Okay, we'll work according to that, no problem. Okay, now coming to, that's all. Hindbrain is over. Okay, now the last part of the hindbrain is medulla oblongata. So this medulla is going to control all the involuntary reflexes. Okay, it has a lot of centers for regulating involuntary functions, like even sneezing, coughing, to any kind of cardiovascular reflexes or gastrointestinal reflexes. Everything can be controlled by the medulla oblongata. Is that clear to all of you? So together all of this makes the hindbrain. Now remember that the brain stem, okay, the brain stem is nothing but the midbrain, pons and medulla. Okay, together these three make up the brain stem. Clear to all of you? That's it. We are done with the hindbrain and we are done with this chapter. Okay, the last chapter that we have to do this evening is chemical coordination and integration. Super simple. I'll teach you what is endocrine glands, what are the hormones, the endocrine system, fat. I will just show you all the different kinds of hormones, the different organs that we have. Remember their function, you're good to go. Okay, then we will talk about the different kinds of hormones that other organs release, and at last we will discuss the mechanism of hormone action. Is that okay? So definitely do subscribe the channel if you have not. Tomorrow morning, I'm repeating myself, we'll be doing genetics. So don't forget to tune in to tomorrow's class. Is that okay? Fantastic, everybody. Tune into tomorrow's class for genetics without fail and subscribe the channel so that you are up to date with all the information. And also join my Telegram channel where I'll be sharing all of these notes for free. You all can utilize. In the description box, we have a link to an AI tool. This is a tool that I created, so you can utilize for infinite practice. It will be very, very important and useful to you. Okay. And also I am doing a study with me challenge. So definitely do tune in every single day into my Telegram or my Instagram and make sure that you are following the challenge. Okay, so that will be super useful to all of you guys. All right, now let's start off with the last and final chapter for today, which is chemical coordination and integration. Okay, so Diya YouTuber, this video is sufficient, ma, because I have covered all the different yes, if you have read it once, it is more than sufficient. So I have covered all the points. Okay, but like I said, you might get statement questions directly from your NCERT, which is why I recommend you to read it once. But if you've already read it, no problem, you can go ahead. So Arjun, we just left with one more chapter, last chapter, and then we can disperse. Okay, so the last chapter here, when you look at chemical coordination, we have the endocrine gland that is playing a very important role. Okay, so endocrine means what? It does not have a duct. Ductless glands. So exocrine is all that has a duct. So all your GI glands, okay, all the different glands that have a duct, your pancreas, your liver, everybody who has a duct will come into the exocrine category. But endocrine glands will directly release into the blood. Okay, and they attack far targets. Okay, it is not like I want to pour the juice here. I want to send this molecule somewhere else where I cannot come in touch with it. Okay, is that clear, everybody? Perfect. So now when you look at the different human endocrine system organs, firstly we have to start with hypothalamus. Okay, then we have the pituitary. Then we have the pineal, thyroid, parathyroid. Then we have the thymus. Okay. Then we have adrenal, pancreas, and the gonads. Okay. These are all the hormones and the different things that you can do. Okay. Now, firstly, here, okay, do you see this structure? This is nothing but the hypothalamus. Okay. Now, the hypothalamus will have two functions. Firstly, it will release either releasing or inhibiting hormones. Okay, so in order to make the pituitary work, pituitary is the master gland, but hypothalamus is the headmaster. Okay, so hypothalamus is going to make sure that the releasing or the inhibiting hormones will tell the pituitary what to do. That's it. Okay, now when it comes to the hypothalamus, it also produces two hormones, which is the ADH as well as oxytocin. Okay, it produces it, but it will not store it. It will give it to the pituitary and ask the pituitary to store it. Okay, then when you look at behind, here we have the pineal gland. The pineal gland releases something called as the melatonin. So melatonin regulates all the sleep-wake cycle. So the fact that night you have to sleep, morning you have to wake up, and this is the time I have to eat, this is the time I have to drink, everything the melatonin is going to take care coming from the pineal gland. Then here we have the master gland, who is the pituitary gland. Okay. Now remember that for the pituitary gland, we have majorly, so for the pituitary gland, we have an anterior pituitary and a posterior pituitary. Okay, so anterior pituitary, if you see, anterior pituitary, I'm just going to write the mnemonic here, okay, which is flat PG. So you can remember, understand all the hormones. F for FSH. So FSH is going to contribute in stimulating the growth of follicles in the women and also regulate the Sertoli cells in the case of the males. Okay, then in case of L, it is the luteinizing hormone LH. So LH is going to take care of ovulation in the case of females, and when it comes to the males, LH is going to take care of the spermatogenesis and the androgen release. Okay, then A is for the ACTH, which is basically adrenocorticoid hormone. So that will stimulate the adrenal glands to release the catecholamines. Then we have T for the thyroid stimulating hormone. So that will stimulate the thyroid to release the thyroxins. P for prolactin, which is involved in milk production in the females, and G for growth hormone that regulates growth in all the individuals. Okay, now apart from that, in the posterior pituitary is where I said it will be storing these two hormones. So ADH is involved in preventing diuresis, which is vasopressin. I already told you when we were discussing excretory products. And oxytocin is going to be the birthing hormone. It is the main hormone that is involved in labor process, okay, and for also milk ejection. Okay, so this is the major functions of these hormones. Now between the anterior and the posterior lobes, we have a central lobe called as the intermedia, pars intermedia we say. So this intermediate lobe has a very important hormone known as the MSH, which is the melanin. Okay. Right, I'm so sorry for the network trouble. I hope it is okay now. Okay, right. So if you see here, thyrocalcitonin is going to reduce the calcium. Okay, then we have the parathyroid glands. The parathyroid is going to release something known as the parathormone. Okay, so this parathyroid hormone or the PTH is going to increase the calcium. Okay, now increase calcium, what do I mean by increase calcium? Wherever possible, it is going to induce the calcium. It's going to take out the calcium from my bones, from my gastrointestinal tract. It is going to help me in all kinds of ways so that I can restore the calcium level in the body. Okay, that is the fundamental role of the parathormone. And then we have two important glands here, which is the adrenal as well as the pancreatic glands. Okay, so the adrenal gland, if you see, first we will look at the pancreas. Okay, now the pancreas is doing dual function. Okay, it's going to do both. One end, it is going to do the it is going to do the exocrine this thing. Okay, just give me 1 minute, guys. Yes, I hope it's clear now. Yeah, so the pancreas have both exo and the endocrine functions, but this part, the in-between part that you see called as the islets of Langerhans, is what is going to have the cells that contribute to the endocrine part of it. Okay, now firstly we have the alpha cells, beta cells and delta cells. So alpha and beta is majorly there in your syllabus, so we will focus on that. If you look at alpha cells, they secrete something called as the glucagon. Okay, so glucagon is going to increase the blood sugar. Okay, whereas beta cells will release insulin, which will decrease the blood sugar. So in case insulin doesn't function properly is when you get something called as a diabetes mellitus, and you cannot process the sugars in your body. Okay, then coming to your adrenal gland. So there you understood glucagon and insulin. Here in the adrenal gland, we have an adrenal medulla that's going to release the catecholamines. Who are the catecholamines? They are the adrenaline and noradrenaline. Okay. Now these both are the fight and flight hormones. So they induce all of these reactions in the body to help you fight or flight for a stimulus. Okay. Then we have three regions of the this thing, which is basically zona glomerulosa. This is going to release mineralocorticoids. Okay. Then we have the zona fasciculata, which will release the glucocorticoids. And we have the innermost reticularis that is going to release the androgens. Okay. Now, mineralocorticoids is for mineral processing, reabsorption in the kidneys and so on. Glucocorticoids is mainly for glucose metabolism, mainly involves cortisols. And androgens mainly include a lot of sex hormones and also a lot of hormones that contribute to the overall growth and development of the body. Okay, they are also involved in reabsorption of the kidneys, and they are also involved in the fight and flight hormones, majorly all the metabolic activities of the body. Clear, everyone? Now coming to the last set of glands. Okay, firstly we have the ovaries. Okay, now in the ovaries we have the follicles that secrete estrogen. Okay, and estrogen is mainly involved in proliferation of the uterus, growth of the uterus and building tissues in the uterus, and also contributes to the secondary sexual characteristics of the females. And then once the ovulation is complete, it forms what we call as the corpus luteum. Okay, now this corpus luteum is going to produce what we call as a progesterone, and this progesterone is responsible for all the maintenance of pregnancy and also contributes to more female sexual characteristics in the females. Okay, now both these hormones come from the ovaries. Now when you look at the testis, this is the male hormone involved in spermatogenesis. So we can see that there is a hormone called as testosterone, or majorly the androgenic hormones, okay, that are coming, and they contribute to the male sexual characteristics through spermatogenesis and to also support all the growth and development of glands and other ducts in the body. Okay. Now, other organs that contribute to hormones in the body include the heart. I already told you they release the ANF when blood flow is too much to the heart. They will decrease the blood pressure and decrease the GFR by releasing ANF from the heart. Okay, right. So, the last thing that you need to understand, guys, is the membrane receptors and how the hormones interact. Okay, so here if you see, fundamentally membranes, the receptors for hormones can be on the membranes. This is when the hormone cannot cross the lipid layer. So any protein hormone, any adrenaline hormone, any hormone that cannot cross the lipid layer will be having the receptor on the in the membrane itself. Okay, now this membrane receptors will majorly take the hormone, okay, they will convert and they will call a second messenger because they cannot go directly inside, and then this second messenger will go and communicate to the nucleus and make changes. Okay, so this will take time, but here especially steroid hormones, okay, or those who can cross the lipid bilayer will directly go to the nuclear receptors and they will directly make changes to the cell. Is that clear to all of you, everyone? Right, Raghav, I'm so sorry, Alan Entertainment. I understand the network is a little glimpsy today, but I'll make sure that doesn't happen for tomorrow. Anyway, I will give you all another revision or a glimpse for these chapters so that you're not lacking anywhere. But nevertheless, please do attend the class tomorrow morning. So tomorrow morning we'll be doing genetics, and I'll make sure any of these network issues do not rehappen.